Ah, oh no. I started. Hello, everyone. Happy Friday and actually, happy summer. Today's the first day of summer. I'm so happy it's summer. Thank you for joining our webinar today. My name is Sumaira. I am a patient living with seronegative neuromyelitis optica spectrum disorder. I was diagnosed 10 years ago, so I'll be celebrating my 10-year anniversary this summer. And 2 months after I got diagnosed, I started the Sumaira Foundation, which is also 10 years old this year, and we are dedicated to raising awareness of NMOSD, MOGAD, and other rare neuroimmune conditions. We're very passionate about building communities of support for patients and their caregivers. We fund research and advocate on behalf of patients. It is my pleasure and honor to say that we are truly patient-led, patient-powered, science-driven, and therapy agnostic. Today, you are joining us for a very special webinar featuring a very special person. So the first thing that I want to say is that 10 years ago, when I was diagnosed with NMOSD, there were 0 FDA-approved therapies. And now in 2024, we have 4 FDA-approved therapies that have shown very positive results for patients living with aquaporin 4 positive NMOSD. So with us today, we are being joined by Dr. Sean Pittock, who is the chair of neurology at Mayo in Rochester, Minnesota. I'm a very big fan of Dr. Pittock. I have been for years. This is our first webinar together, so I'm really excited. But Dr. Pittock and I have done something else together. We were actually in a documentary a couple of years ago that aired on PBS. I will share the link in the chat. If you haven't yet watched it, please do. It's called Blindsided Within. Fair warning, I'm a bit of an ugly crier and it's a lot of tears in there, but I'm sure many of you can relate. But please go ahead and watch that documentary when you have a chance. And just a couple of housekeeping items. So this webinar is being recorded and it will be available for replay on the Foundation's YouTube channel as well as our global website. There will be a dedicated section or time rather for questions. So don't be shy. Go ahead and populate your questions in the chat and the Q&A, and we'll get to as many as possible. And a very special thanks to our sponsors for making this program possible. That is Amgen, formerly Horizon, and of course, Alexion. So without further ado, please join me in welcoming Dr. Sean Pittock.
Well, thank you very much, Sumaira, for inviting me. I'm really looking forward to not only talking about the great strides that have been made in aquaporin 4 antibody positive NMOSD, but I think that people that do or do not have this condition, this really opens the door for great advances in all forms of inflammatory CNS neurological disorders. So I'm going to talk about approved NMOSD treatments, but I'm going to start really and just tell you really the story of really how this all started. So as many of you will know, NMOSD was considered before the discovery of the aquaporin 4 antibody a very severe form of multiple sclerosis. Such a severe form of multiple sclerosis that it could cause patients to become blind. 50% of patients would become blind in 5 years. 30% of patients could die within 5 years. And about 50% of patients could end up in a wheelchair within 5 years. So this is like probably the most severe form of multiple sclerosis. It was actually called optic spinal multiple sclerosis in Japan. But something really changed, and it changed in 2004 and subsequently in 2005 when this person, Van der Lennen, who's really a pioneer in the field of neuroimmunology, discovered that patients that had the NMOSD phenotype, this very severe form of MS, that 70% of the patients, when she took their blood and put it on mouse brain tissues, she saw that they had antibodies that bound to the mouse tissues with this staining pattern. And we had tested hundreds of thousands of patients, their bloods for these antibodies, and we never saw them. So we knew that this was special, and we knew that this was biomarker. And we subsequently then discovered that it was targeting a water channel on the outside of an astrocyte. And this was a seismic change in thinking in MS research. And in fact, this paper, which was the first ever biomarker of an MS-like illness, got hit with a lot of resistance because it was pushing the limits, and it was basically moving us from main focus on the oligodendrocyte to actually another cell in the brain called the astrocyte. And this discovery really changed at least a lot of the thinking, because having the biomarker really allowed us to define the disease entity. And we suddenly realized that NMOSD was actually a much more complex disorder than we had previously thought, with a much broader imaging findings. Now, as you all know now, we know a lot more now than we did back then. We know that there are three major types of attacks that can occur in NMOSD. The first one is optic neuritis, where you get inflammation of the optic nerve. The second type of attack you can get is transverse myelitis, where you get a long lesion in the spinal cord, and patients can have weakness, bladder problems, difficulty walking. And then the third is the area postrema syndrome, where patients can have intractable nausea and vomiting, where they just keep vomiting and keep feeling nauseous, sometimes even up to a year. So these are the characteristic attacks that can occur in an NMOSD. Now, you'll all know the standard treatment approaches to an attack in NMOSD is intravenous methylprednisolone, plasmapheresis, or intravenous immunoglobulin. But we're not going to be focusing on how we treat attacks. We're actually going to be focusing more on how to prevent attacks. But I do just want to share this with you. This is studies that we did back in 2016, led by Owen Flanagan from our group. But we did this in collaboration with Philippe Cabret, because we were interested in looking not only at how common is NMOSD in white North Americans, but how common is it in Africans and people of other ethnicities. And so what we did was we studied it in the Caribbean, in the French West Indies. And what we found was very interesting. Essentially, 3 people out of every 100,000 whites have NMOSD. But it's much more common, about 3 to 4 times more common in Afro-Caribbeans, about 1 in 10,000 people. And overall, about 500,000 people have NMOSD at the current time. So it's not an insignificant disease entity, given the seriousness and the severity of the illness. And as you know, it affects women much more commonly than men. And it generally comes on in the 30s to 40s. Now, the important thing about NMOSD is the big problem with NMOSD is that the disability is caused by the attacks. This is very different to multiple sclerosis. In multiple sclerosis, yes, you have clinical attacks, but they get better. And most of the disability in multiple sclerosis comes from the progressive phase of the illness that occurs in the 50s. In NMOSD, we don't see a progressive phase of the illness. The disability is attack related. And so this just emphasizes the importance of stopping the attacks. If we're going to make an impact in this disease, we have to stop the attacks. Now, this is the old treatments we used to use. Some of you may be on some of these treatments. Rituximab is probably the most common treatment that's currently used. And you can see these are just some older studies looking at, what % of patients are free of relapse. You can see that over varied follow-ups, we do see people having breakthrough attacks with some of these drugs. Azathioprine, for example, was one of the drugs we used in the old days, but you can see that we have a lot of breakthrough attacks here with that drug. About 40% of people will have breakthrough attacks on CellCept, and the Rituximab data ranges from 20 to 40% of patients having breakthrough attacks in their follow-up. So a breakthrough attack is really not acceptable, because you can have disability from an attack, and so we really need to stop the attacks. Now, if we look at how things were when we had access to those older drugs, you can still see we had some problems. So 57% of people were losing vision after 5 years of diagnosis in an eye. 34% of people were having some problems with motor function. 23% were needing a wheelchair after 6 years of diagnosis, and some patients were still dying. So this just reiterates the importance of getting some new drugs. And so this just shows you how all the work that started to happen once the water channel antibody was discovered, we started to begin to tease out all the ways in which those antibodies cause disease. How are they made? How do they access the water channel? What effects do they have on the water channel? And we identified that interleukin-6 was a protein that would drive the production of antibody. And the antibodies were made by B cells. And those B cells had CD19 on their cell surface. And we also noted that when the antibodies bound to the target, the water channel here, it activated complement, and the complement would kill the neural cell. And other proteins like C5a would cause massive attraction of inflammatory cells into the brain. And so these gave us some opportunities for possible therapy. We could block interleukin-6. We could kill any cell that has CD19 on it, or we could block complement activation. So this led to some trials, and I'm going to tell you a little bit about the trials that led to the availability of many of these drugs to patients with NMOSD in the USA. So the first trial was the PREVENT trial. This used a drug that blocks complement. Here you can see it's a C5 complement inhibitor. And this was a randomized double-blind time to event study in 143 patients. And the patients were blinded to the drug. So they were allowed to receive their standard of care. That could have been low-dose prednisone with Cellcept or with azathioprine. They couldn't have received Rituximab in the preceding 3 to 6 months. And the patients that received the Eculizumab received 900 milligrams intravenously weekly for 4 weeks, and then 1,200 milligrams every 2 weeks after that. And patients that are on Eculizumab now, they receive the medication in this way. And what you can see is that there was a dramatic effect here. So in red, this is the attacks, the likelihood of having a clinical attack if you were on a dummy drug compared to Eculizumab. And you can see that about half of the patients on the dummy drug in the trial had a clinical relapse, but only 3 patients taking the Eculizumab in the trial had an attack. And so that was a dramatic effect. In fact, an effect that was never seen in the MS literature. But what we found was a 94% reduction in the likelihood of having a clinical attack if you were on Eculizumab in the trial. And not only that, as you followed the patients out 2.8 and now 5 years, that benefit persisted. In other words, they continued to be free of clinical relapses. Now, what about some other drug trials? NMOmentum was a trial, a placebo trial of Ravulizumab. Now, Ravulizumab is very like Rituximab. It essentially kills B cells that have CD19 on them. So that's essentially all the CD20 positive cells, but then in addition, some additional cells that lack CD20. And these cells, when they're killed, they potentially reduce the production of the bad antibody, the aquaporin 4 antibody. Now, the thing that's interesting about this study is it had a pure placebo arm. So at the time, there was a lot of controversy about whether it would be ethical to have somebody go on no drug. And so because of that, the design of the trial was a little different. We had a much shorter duration of time that the patient was allowed to go on no drug. So the max was 6 months. And the patients were randomized in a 3-to-1 fashion. So if you went into the study, you had 3 times a likelihood of getting the drug than the people that ended up with placebo. But again, the study outcome was quite dramatic. You can see the patients on the placebo were much more likely to have a clinical relapse or attack than the patients that received the drug during the trial. And so you can see in the NMOmentum trial, 77% risk reduction in the likelihood of clinical attack in the patients that received the drug and were aquaporin 4 antibody positive. In this study, they also did include patients that were seronegative for NMOSD, but we did not see a benefit in those patients, though there was only 17 of them. And the other interesting thing about Inebilizumab is that in the extension trials, as we go out over 4 or 5 years, the drug has a persistent benefit. So the drug really works and continues to work over the longer term, which is very exciting. This just shows you here the disability outcomes in this trial. This trial was really the only trial that showed evidence of reduction in disability when you measure it by EDSS. And there's many reasons why that may be, but just to point that out to you. Now, here are the 2 trials of Satralizumab. So the Sakura Sky trial. This is an IL-6 blocking agent. You might remember that I showed you that interleukin-6 at the time of an attack increases in the spinal fluid, and that drives the B cells to produce the aquaporin-4 antibody, and we don't want that. And so in this study, you can see the patients received 120 milligrams subcutaneously of Satralizumab at time 0, week 2, and week 4, and then they received it subcutaneously every 4 weeks after that. And that's how you receive Satralizumab in day-to-day clinical practice. You can see here that the patients again had a benefit. Here you can see the likelihood of a clinical relapse on Satralizumab and the much higher likelihood of having an attack if you received placebo. And similar findings with the Satralizumab in the Sakura Star study. So for the Sakura Sky and Sakura Star, just to kind of give you an overview, in the Sakura Sky study, there was a 79% risk reduction in patients that were aquaporin-4 antibody positive, and there was a 74% risk reduction for the Sakura Star study. In the open label extension, we still see significant risk reductions in terms of the likelihood of having a clinical relapse, but it's dropped to 66% over the longer term. Now, many of you will be on Rituximab. Unfortunately, we don't have powered randomized controlled trials of Rituximab. This was an underpowered trial of Rituximab, and the population of patients studied in this were different to the population of patients that were studied in the other trials. But still, in this study, none of the patients treated with Rituximab had a relapse, compared with 7 of 19 patients in the placebo arm who did have relapse. So just to kind of give you an overview of the currently used treatments in NMOSD, Rituximab is not FDA approved because, remember, the trial was not designed or powered in such a way that we can get definitive data. But certainly Rituximab, we know, does reduce the likelihood of clinical relapses and is used frequently in clinical practice. And is certainly a very reasonable drug to use in countries where it's difficult to access these expensive FDA-approved drugs. And it's interesting that if you move even into the developing world, the World Health Organization is working at the current time to approve Rituximab treatment availability for patients from poorer countries where they potentially could benefit from this drug in the context of NMOSD. But in, for example, the USA, the Eculizumab, Inebilizumab, and Satralizumab are FDA approved. And here you can just see the different inclusion criteria, the different designs of the trials, whether they were randomized controlled trials, how long they went on for, what the treatments were, whether they were pure placebo, as in the Sakura Star and the NMOmentum trial, or whether the trials allowed you to use the drug in the context of standard of care therapies. And then the open label periods. Now, I want to talk to you a little bit about a new drug that's just been FDA approved called Ravulizumab. So, as you know, with the Eculizumab drug, we saw this dramatic 94% risk reduction. So that means that you're 94% less likely to have a clinical relapse than if you don't take the drug. And this study was a study of Ravulizumab. Now, Ravulizumab is very like Eculizumab, but the drug has been manipulated so that it has a longer half-life. In other words, the Eculizumab has to be administered every 2 weeks, but Ravulizumab only has to be administered every 8 weeks. So this is a huge convenience benefit for patients that want to be or choose to be on a complement inhibitor. Now, this study couldn't be a placebo-controlled trial because it would be unethical to do a placebo-controlled trial, in other words, you receive a dummy drug, when we already knew that eculizumab worked wonders for the disease. And so this was an open-label trial where we knew what the patient was receiving. This is the way the drug was dosed, different to eculizumab. It's weight-based dosing, where the patients get a weight-based intravenous dose on day 1, and then they get another weight-based intravenous dose on day 15, and then they get that dose every 8 weeks. And that's how it's done in clinical practice. And this was an incredible outcome because in the trial over 75 weeks, and now we have open label extension data up to 138 weeks, we have no relapses in the patients that received Ravulizumab. And we compared that to the placebo arm of the PREVENT trial, because we couldn't have a placebo trial arm in this trial, and you can see the dramatic difference. So this results in a 98.6% reduction in the likelihood of having a clinical relapse with Ravulizumab. So very exciting. Now let's talk a little bit about side effects and some of the concerns that some patients have and that came through from these studies. But I also want to draw your attention to the fact that eculizumab has been used in many other diseases. And so we know essentially what the risk profile of this drug is, as well as Satralizumab, from learning about side effects that occurred in other diseases for which the drug is used. So, for example, eculizumab is used in the treatment of myasthenia gravis. It's used in the treatment of paroxysmal nocturnal hemoglobinuria, and it's used in the treatment of transplant rejection. What we saw here was that overall, the side effect frequencies in the patients that received eculizumab were very similar to the patients that received the placebo or the dummy drug. You can see that there were some more upper respiratory tract infections in the eculizumab treated patients. In the PREVENT trial, there were no meningococcal infections because meningococcal infections are a risk if you have complement inhibition. And it's a big risk. Patients that have or on eculizumab have a card that they must carry on themselves at all times. And if they get any symptoms of meningococcal infection or meningitis, they must go straight to the emergency room. And the risk of meningococcal infections with eculizumab is about 2,000-fold higher than the general population. In the study, the PREVENT trial, there was one death, but it was felt to not likely be related to eculizumab. And then we had good tolerance with the long-term treatments. With inebilizumab, again, similarly, a good side effect profile, similar side effect frequencies when you compare those that received inebilizumab with those that received placebo. But you see there was some more arthralgias, 10% versus 4%. But there were similar rates of injection site reactions and similar rates of infection. There were 2 deaths that occurred during the open label extension. One was relating to disease worsening in the context of a chest infection, and the other one was an indeterminate brain lesion. The treatment was generally well tolerated over the 4-year treatment trial period. And then with Satralizumab, looking at both of the studies combined, similar again adverse events when you compare the patients that received the drug with the placebo. And this is just because any symptom that a patient might have in the context of a study is reported. Again, you can see similar rates of serious infection, no anaphylactic reactions, and there were no new safety signals compared with the other diseases that are treated. This just gives you a sense of all the trials and just the big number. But here what you can see is the total number of adverse events in the placebo and the treatment for PREVENT, in the Sakura Sky, again, placebo and treatment, in the Sakura Star, placebo and treatment. And you can see the numbers are quite similar. We're not really identifying any major difference between the patients that receive placebo and treatment. Now, I do want to draw your attention to these 2 patients. In the Ravulizumab trial, the Champions trial, where we had essentially 100% freedom from clinical attacks. In other words, we had no clinical relapses. In that study of 58 patients, there were 2 patients that did develop meningococcal infections. This is the first patient who developed meningococcal infection. They had been vaccinated, and they were not on any concomitant immunosuppressant therapy. And they were treated rapidly and made full recovery, but they decided not to continue in the trial. And then the second patient, also vaccinated, but was on a lot of other immunotherapy, potentially could have impacted their vaccine response. But they did develop infection 482 days after the Ravulizumab was started. And you can see that they were treated rapidly with antibiotic therapy, and they made full recovery, and they did stay in the trial. Now, I wanted to bring your attention to that. This is something that's going to be monitored in patients that go on Ravulizumab, and this is an important issue to be addressed. And patients must be informed of these risks and must understand the importance of acting quickly should they develop any symptoms of meningococcal infection. Now, what are the kind of factors that would influence a patient deciding on which drug to try if they have access to these drugs? Well, obviously availability. In the US, we're lucky because patients do have access to these drugs. I have not had a situation where a patient has not ultimately got on the drug that they and we decided was appropriate for them. Again, we need to make sure that we have the right diagnosis, because remember, patients with seronegative NMOSD did not seem to benefit from Inebilizumab or Satralizumab. So patients have to have access to good quality testing for aquaporin 4 antibodies. And patient preferences are really important. There's a lot to be said about receiving a medication that you just have an IV infusion every 6 months versus, for example, Eculizumab, where you have to have an infusion every 2 weeks. And these convenience issues are important for patients, especially patients that have very active lives or are working or are trying to raise families. So this just gives you an overview of efficacy. I just did this in terms of risk reductions, complement inhibition in blue, interleukin-6 inhibition in yellow, CD19 inhibitor in purple, and then Rituximab or CD20 inhibitor in black. And you can see the varied risk reductions and the statistical significance. Now, before we finish and take questions, I wanted to just introduce this study that I was involved with. It was published in 2019 with Richard Burt as the lead author, and it looked at hematopoietic stem cell transplantation for the treatment of neuromyelitis optica. And I wanted to bring this to your attention because here you can see that we reported quite a dramatic and good response here. There were patients that went from seropositive for water channel antibodies to seronegative. So we switched off and got rid of their antibodies. And a lot of the patients, about 80% of the patients, remained relapse-free out to the 5-year follow-up time point. And in that paper, we suggested that this was really a good option, potentially, for patients with NMOSD. Now, unfortunately, many of these patients, after the 5-year time point, they kind of got lost to follow-up. But I came in touch with a Mayo Clinic patient of mine, and subsequently also working with colleagues down at UT Southwestern, we identified another patient who, at the 10-year time point, post-transplant, developed clinical relapse and attack. And in fact, one of the patients, unfortunately, had such a bad attack that they developed a complication and died. And what I want to show you here is that this was the original study where we showed that the antibodies kind of came down and went to negative, as you can see. But then when we looked at these 2 patients that we are actually going to be presenting at the ECTRIMS and AAN in summer, these 2 patients, you can see, they got the transplant, patient 1 and 2 in red and blue, and look, their antibodies dropped to undetectable, and they had no clinical relapses. But look, after 10 years, the antibodies came back up, and they had clinical relapses. And I just wanted to point this out, that if you're having or thinking about a stem cell transplant, it's not proven in any kind of randomized trial fashion. But the take-home message here is you must follow up with your neurologist every year, and you must make sure that you're checking those antibodies. And if those antibodies come back, you should start one of the disease-modifying agents we've talked about. I just want to bring that to the attention of the NMOSD community. So it's been a roller coaster for me. Essentially 20 years of my life been spent working on this. At Mayo Clinic, Brian Weinshanker and Dean Wingerchuk, who many of you know, defined NMOSD. They defined the criteria. Then Claudia Lucanetti looked at the brain spinal cords of people that had had biopsies, because sometimes people are misdiagnosed with tumors when they go in with NMOSD. And she saw that there was complement in the lesions. In 2004 and 2005, Vanda Lennon at Mayo and our group discovered the water channel antibody. Then internationally, everybody hopped on board. Publications went from 3 or 4 a year to thousands a year. We went from a few investigators in a few countries interested in this disease to thousands of people interested in this disease. We figured out the immunopathogenic mechanisms. We identified novel targets. We then went to drug trials. And in 2019, we had the year of NMOSD, where we had 3 FDA-approved drugs. And then most recently, in 2023, I've just told you the Ravulizumab with 0 attacks. So very exciting in this field. We have a lot of work to do. We have all of the autoimmune encephalitides. We have no phase 3 trials in any of those diseases. We have the whole field of seronegative encephalitis, seronegative NMOSD-like illnesses, seronegative recurrent optic neuritis, seronegative recurrent longitudinal extensive transverse myelitis, all of these conditions for which we have no treatments. So we have a lot of work to do. But we can use this as the paradigm to ultimately attain success. So early diagnosis is key. Improved understanding is key to discovering new drugs. I've talked about biomarker discovery to near cure. This experience can be the basis for how we design trials and move forward in the future. And there's great hope. We can essentially nearly cure this disease, and hopefully you can have access and gain access to some of these drugs. Thank you very much.
Wow, that was incredible and very inspiring, Dr. Pittock. It was outlined very nicely, so thank you for that. We do have some questions, so let's just get right to them so we can answer as many as possible. The first question is, I'm getting ready to switch from Soliris to Ultomiris. I've done well on Soliris, no relapses in 5 years. My only desire to switch is time frame so that I can go from 2 weeks to 8 weeks. I'm concerned about duration between treatment. Should I have a backup plan? Should I encounter NMO issues during the new time frame?
Yeah, good question. No, I think the way you should do it is, you need to coordinate it with your physician so that when you get your, say you get a dose of Eculizumab, you know that your next dose needs to be in 2 weeks. Well, in 2 weeks, within that time frame, within 2 weeks, you want to get your first dose of weight-based Ravulizumab. Then you remember at day 15 after that, you get your second dose of Ravulizumab. And then after that, you're just on the every 8 week. And I don't think you have any concerns because we've studied the pharmacokinetics. That means that studying the concept that you have to have, by dosing that way, the right amount of drug in your system to prevent you getting a clinical relapse. And we know that if you follow those instructions, you will be protected. But going to an 8 week is great because it opens up your opportunities. So, for example, many patients are limited. They can't go on a vacation for 3 weeks if they're on Eculizumab. Now you can go on a vacation for 8 weeks. So that's good. It opens up opportunities. Remember, with Eculizumab, you cannot miss a dose or be late for a dose because this happened in COVID. Patients, in fact, we've had patients where the doctors stopped the medication during COVID and the patients had clinical relapses. We had situations where patients missed or were delayed getting their infusions because of COVID and they had relapses. So You definitely have to make sure you get your drug every 8 weeks, but it does open up convenience for you and opportunities, and reduces the time that you have to go down to the infusion center. So it's good.
All right. Next question. I have just started on Ultomiris. Can you reassure me that I am taking a safe drug and not part of the effort to confirm its safety? I would say you already answered this.
Yeah, I mean, I will be honest. The risk of meningococcal infection is real, but there is no drugs that don't come with risk. I will, So let me tell you a few things. So first of all, the most important thing when you're starting on a drug like Ravulizumab is that the patient is really engaged, that you're really engaged in your care. There are some patients, to be honest, that I would not recommend that drug for. Patients that are non-compliant, they miss doctor's appointments, they don't turn up, they're late. That's not the right patient to have on the drug. So if you're really motivated and you're really involved in really maintaining your health, you need to be alert. If you get headache and fever, you need to go to the emergency room. You need to have that card with you. We also have an international group that we're currently working with, and this is done external to the drug companies, though we do have on the committee, they're not voting members, but we do have representation of the drug company because they know a lot of information. But we have convened experts, for example, like Michael Yeaman, the world's experts in meningococcal vaccination, Ray Burrows from the UK, the world's experts on a variety of different aspects of meningococcal infection. And we're going to be coming out with a delta analysis or a RAND analysis of expert opinion regarding how we would best mitigate the likelihood that you could have an infection. Unfortunately, it's very difficult because the data is actually really problematic. But at least it will be an expert opinion, and hopefully physicians will follow that. That's the major issue with Ravulizumab. And yes, efficacy, you're on the most efficacious drug. But yes, there is that side effect, and we have to watch you carefully, and you have to watch yourself carefully.
Okay. Can you share any long-term outlooks for Ultomiris?
Well, I mean, I think the long-term outlook is that it'll be the two blips we saw will dissolve away into what we're seeing as the standard side effect profile for Ravulizumab across all indications. So, for example, we did not see an increased risk for meningococcal infection in myasthenia gravis patients being treated with Ravulizumab, in patients treated with all the other diseases that eculizumab had previously been used for. And so we're just hoping that we won't see this as a problem. I think from my perspective, from an efficacy perspective, it's a very, very impressive drug.
What about patients planning pregnancy or those who are pregnant?
Okay, that's a good question. And it's a good question because remember, the ratio of women to men is 9-1. And the age group that people get this disease is in fertile years when you can have children. So in terms of management, first of all, the data, no drug is approved for pregnancy. That's the first thing. But we do know that eculizumab seems to be safe in pregnancy. And we know that because many of the children with PNH who are still alive because of this wonder drug, many of those patients, they have to get the drug during all the time. They can never stop eculizumab. And many of those have gone on to have children and have had successful births, and we have not seen an increase in abnormalities in the babies, at least as of yet. So that seems to have a reasonable safety profile, but it hasn't been given a designation of safe in pregnancy. But I think you could breathe if you are on eculizumab in the context of pregnancy. Now, Satralizumab, we don't have good data on. Inebilizumab, we generally tend to take the same approach as we take with MS. So, for example, remember, inebilizumab is a drug that's given every 6 months, and it actually lasts maybe for a year. So you could have a situation where you get an Ravulizumab infusion, wait for a couple of months, then get pregnant if you can, and then don't redose until you deliver. You still have that protection over that period of time. And that's probably a reasonable approach. And also remember that steroids are somewhat safe in pregnancy as well. So it's complex. It's an area that you certainly need to try to preempt, try to work with your physician before you get pregnant, try to do it in a planned way, and try to use the information that you have and the physician has to the best of your ability to optimize the outcome of the pregnancy and reduce the potential risks to the fetus.
How do these preventative therapies compare in terms of their impact on persistent symptoms that patients may experience between relapses, such as pain or fatigue?
Good question. So these drugs will not help your symptoms. The only thing that these drugs will do they reduce the likelihood of you having a clinical relapse. So if you have pain, if you have fatigue, if you have tonic spasms, if you have a variety of different symptoms, these drugs will not benefit those. We had thought there was some literature indicating that IL-6 inhibition may help neuropathic pain, especially from spinal cord injury. And there were some reports in the literature of case reports of patients starting Satralizumab and noticing improvements in their pain. But in the phase 3 trials, we did not see that as a benefit. So at the moment, I don't think that there is good evidence that these drugs help those day-to-day symptoms. Those symptoms can be managed with alternative approaches, symptomatic approaches, biofeedback, acupuncture, a variety of different neuropathic pain meds, those types of approaches. And there are some patients, I have patients that tell me, I know my symptoms are coming back before I get my next infusion. So I don't have a good mechanistic explanation for that.
Is disability improvement an important issue for choosing a medication?
That's a good question. Not really. So the medications that we've talked about, the preventive medications we've talked about, are really preventive. So they aren't going to impact disability. Now, obviously, if you've had a recent clinical relapse and you have disability from it, you have a window of probably a year during which you can make good recovery. But the drug itself won't impact that recovery. Now, in terms of disability, the main approach is stop the clinical attack, stop disability. But what about people that have had this disease for some years and now are disabled? What's their opportunities? So at the moment, other than our symptomatic therapies, the types of things that are going to impact disability in the future are going to be the following. So there's a lot of work being done on the possibility of stimulation, of electrical stimulation, the use of electricity to help patients that are having neuropathic pain, that are having gait problems, that are having spasticity. In fact, we just recently put electrical stimulators into the spinal cords of 2 patients with progressive multiple sclerosis. And we're currently studying the potential benefits of that. Neuroregenerative trials looking at regenerative medicines like stem cell, mesenchymal stem cells, those types of potential therapies. And we're really kind of talking about regenerative medicine here. But we don't have anything at the current time, right at this point, for that.
I have a question for me. You've now been in this field for so long, and you've obviously seen the evolution of NMOSD from where it was before, which was a very devastating condition. And in some cases, it still is today, but the prognosis is better and we have more options. So my question is, what excites you about the future of NMOSD? What kind of keeps you going? And you mentioned being very interested in autoimmune neurology in general. So can you just tell us what you're excited about in the future?
Yeah, I mean, I think, well, first of all, for NMOSD, it's going to be difficult, at least for aquaporin 4 antibody positive NMOSD, to really move forward into new drugs. So what would be kind of exciting new drugs to think about? Well, the possibility of a vaccine would be very exciting. The potential of vaccinating someone and turning off or switching their immune attack to a tolerance or a tolerogenic immune process. And the second would be the potential use of CAR T therapies to knock out CD19 positive B cells. This would be potentially a treatment where you might only have to get it every 4 to 5 years. The problem with that, though, is you can imagine and you can hypothesize that that would be great to do, but it's going to be difficult to do that study in a country, for example, in the US, where people have access to a drug that will reduce the likelihood of you having a clinical attack by 98%. So how would you do that study? How would you design the study? What are you going to use as your comparator? Now, in the field of all the other diseases where we don't have any FDA-approved drug, the time is ripe for all of these different approaches. So the first thing we need to do is we need to try to understand mechanism. So if you take people that have seronegative disease, how are we going to, first of all, find biomarkers that, like water channel, really provide us insights into what's happening in that disease? How is the disease occurring? So that if we can figure that out, then we can identify targets that will stop the disease occurring. And so there's lots and lots of opportunities here, but the technology is advancing dramatically. So, for example, the potential of developing precision medicine, where, for example, you just use an antibody that just blocks that antibody, no other antibody, just blocks that antibody. You reduce the side effect profile. You are not using a blunt tool like knocking out all 85, 90% of the entire B cells in a patient, or blocking the entire complement, or knocking out the stimulation for all plasmablasts with IL-6. The possibility of doing precision medicine where you just take out the bad B cells that are making the specific antibody that's causing a problem. And you could do that with CAR T therapy. You could design a CAR T that just takes out that bad B cell. And the same with T-cell mediated diseases. You could design CAR T that just takes out those bad T cells. So I think the future is bright, but it will require a lot of investment. And the people that have most of the money are the drug companies. And so they're going to be trying to move with their drugs. And that's great. That brings excitement. But in order for the success of the field, if the patients don't engage in the drug trials, the drug companies will lose interest and we won't have anything, because these are rare diseases. So for the Sumaira Foundation, for all other foundations, to really promote patients to get involved with drug trials and become a part of the solution. So there are patients that I had in my original trials, patients that joined us in our kind of biorepository efforts to understand disease. And many of those patients either entered into the trials or are now on those drugs. So, get involved, become a part of the solution, I think is really important for organizations like the Sumaira Foundation, but then also patients themselves.
Absolutely. That's a great message. And it's something we try to share with our patient community all the time. We can help the collective cause by getting involved and engaging in these trials that will help us, that will help doctors, that will help research, that will help science, that will help the future. So I am with you on that. Back to the questions. If I also have lupus and I am on other immunosuppressants for that as well, should I worry about the efficacy of my meningitis vaccine while on Ultomiris?
That's a really good question. So let's just clarify for the other members of the audience what we're talking about here. So it relates to people that have NMOSD and say have been treated with Rituximab and have failed, or are transitioning from Rituximab onto a complement inhibitor. And now what's happening is, whether you have lupus, rheumatoid arthritis, or any immune-mediated disorders, and you're already immunosuppressed as a treatment for those other conditions, and now you're going on to a drug for your NMOSD, you're vaccinating the person when they're immunosuppressed. So their vaccine response is going to be really, really attenuated. We know this from COVID, because we know that people that were immunosuppressed when they were getting their COVID vaccines had a significant reduction in their response, in their immunological response to the vaccine. So you're absolutely right. This is an issue. So what do we do about that? And that's one of the issues that the international group that I'm working with is trying to address. But let's talk about how we might address that. So first of all, the reality is that most times people have meningococcal infections. They've actually been vaccinated with the serogroup that actually now they've been infected with, and they were vaccinated when they were not immunosuppressed. So being vaccinated in a non-immunosuppressed state still doesn't protect you 100% from getting meningococcal infection. The other possibility is that you could imagine that, say you were on Rituximab and now you've just started Ravulizumab and you know your vaccine response wasn't very good. You might want to wait, in other words, revaccinate when the CD20 count has come back up. In other words, the Rituximab effect has worn off. The CD20 count has now come back up. You're still on your complement inhibitor for your NMOSD, but then you potentially could revaccinate at that point. And the other question, and this is not addressed, but I'm just throwing out some ideas here to you, is whether or not people that were immunosuppressed at the time that they were vaccinated and are on a complement inhibitor, should they be on chronic maintenance antibiotic therapy, prophylaxis? So those are the kind of things that we're grappling with at the moment. But that's a very good question and something that's really on the minds of the physicians as we try to navigate this question with limited data to support our decision making.
Okay. Dr. Pittock, when do you consider switching NMOSD patients to another drug? Of course, I presume a relapse would trigger that discussion, but are there other reasons patients switch therapies? And if so, could you describe those situations?
Okay, so there's an easy situation. So you're on one of the older drugs and you have a relapse. That's easy. You absolutely should go on an FDA-approved drug. Now, then the question is, how far back do you need to go to say you should switch? So for me, it's probably 2 to 3 years. I mean, if you're on CellCept for the last 15 years and you haven't had a clinical relapse, I don't know. That's a difficult question. Now, there's some patients that will come and say, I read these drugs, I read their efficacy, I want to go on the drug. So the patient is leading that discussion. And in that situation, I go with what the patient wants to do. But a lot of patients are on Rituximab. They've been on Rituximab for 10 years. They haven't had any clinical relapses. They feel comfortable on the medication. I think that's reasonable for them to stay with what they're comfortable with. But for newly diagnosed patients, I think it's incorrect, not good medicine for a newly diagnosed patient to not go on an FDA-approved drug. I think that's the wrong thing to do. I think they should be put on an FDA-approved drug.
I was diagnosed with NMO a year ago, but I am not taking any medication or drugs. My neurologist has not mentioned anything about these drugs to prevent relapses or attacks. Should all NMOSD patients take therapies for NMOSD?
Okay, so let's talk about NMOSD. What I've been talking about today is generally has been aquaporin 4 antibody positive NMOSD. The drug trials that we've just discussed only relate, in other words, those positive benefits only relate to people that have aquaporin 4 antibody positive NMOSD. If you have aquaporin 4 antibody positive NMOSD and you are not on one of those drugs, then you are being mismanaged. I consider it criminal if you are not on an attack-preventing medication. We know that from the natural history of the disease. Now, supposing you have recurrent optic neuritis, or you have had an attack of optic neuritis and you have a lesion in your spinal cord, or you have another multifocal inflammatory brain disease that's not NMOSD, aquaporin 4 antibody positive, then it's difficult because we don't have the data. So, for example, if I saw a patient that has relapsing optic neuritis and they're not MS and they're not aquaporin 4 antibody positive NMOSD, the first thing to do is check them for MOG, because they could have MOGAD. But recurrent optic neuritis patients, we know that 70% of patients are negative for all antibodies. In those patients, I do treat those patients if they're having relapsing optic neuritis, but I don't use the drugs we've just been talking about. I'll use drugs like Cellcept, Rituximab, prednisone, sometimes azathioprine. Sometimes I'll do things like measure cytokines, and if the IL-6 is really up high, I might use an IL-6 inhibitor. But in that situation, I'm really working with the patient in an individualized approach to come up with the best treatment for the patient. But if you're a patient that, say, just had one episode of optic neuritis, if you're aquaporin 4 antibody positive, you're on lifelong attack-preventing therapies. But if you're not aquaporin 4 antibody positive, and even if you're MOG antibody positive, I wouldn't put you on an immunotherapy. I would watch you. So it's very individualized.
Given that seronegative patients did not seem to show benefit on Inebilizumab and Satralizumab, which treatment approaches do you use for seronegative patients? Do you think there will be any treatment trials in the near future that will study this particular population?
Okay, well, first of all, in the momentum trial, a third of the patients that were seronegative NOSD were MOGAD. Okay, so now we're in a situation where we now only had 7 patients that were MOGAD, and we had 10 patients that were a mixture of, we don't know. They had a phenotype that looked like NMO, but they didn't have either of those antibodies. And maybe within that group, there's 4 or 5 different diseases. So we have to understand and discover what those diseases are in order to understand how to treat them. Now, if you have a disease that is has occurred once, we don't know if it's going to recur again. So, for example, MOGAD patients, half of them don't ever have another relapse. So we would be harming those patients if we put them on a disease-modifying agent, if we suppress their immune system over the longer term, knowing that half of them are never going to have another relapse. That would be the wrong thing to do. So you have to look at the patient. If the patient is having a relapsing inflammatory brain disease, then you have to try and prevent that if it's immune mediated. And so then you use the drugs that we used for NMO before we had the FDA-approved drugs. You use those types of approaches.
Okay. Do you consider continuing Rituximab during pregnancy, or start or switch to Rituximab for pregnancy planning according to MS experiments?
It's something that you could do. Again, Inebilizumab is the drug that's FDA approved for NMOSD. So generally, we would use that in a similar way as the MS physicians use Rituximab in the context of pregnancy, or Ocrevus in the context of MS and pregnancy.
We have 2 more questions. If you are on Rituximab for less than 1 year and want to switch to the new therapy Ravulizumab, but haven't been vaccinated for meningitis, how can you switch and get vaccinated before changing to the new 8-week drug?
Well, you just get vaccinated and switch. But yes, your immunological response to the vaccine will be attenuated. It will be weakened because you're on the Rituximab. And that's why I was saying you get vaccinated. Maybe in this situation, your doctor might want to cover you with an antibiotic prophylaxis until your B cells come back and you're revaccinated. But we don't, this is not a guideline. This is a conversation that has to be had.
Dr. Pittock, oftentimes I hear from patients who are on Rituximab but curious about the new therapies that are out. They often express that they're like nervous to bring it up to their doctor because the doctor hasn't brought it up to them. So, as a treating physician, can you offer some advice to patients who are watching this? Like, what's the best way to bring it up to your doctor if they haven't yet brought it up?
Yes, advocate for yourself, fight for yourself. Bring the information from the Sumaira, from the foundations to your doctors. It's not good enough for a doctor to say, I don't know what this disease is. I haven't heard of it. You fight for yourself. We live in a health system where you have to advocate for yourself or your loved one.
Absolutely. I don't think I could have ended that on a better note. Everyone, thank you so much for joining. This was incredibly informative. And Dr. Pittock, you are obviously incredible. We loved having you. So thank you. I hope this was enjoyable for you too. And for anybody who is watching this after the fact, this is, going to be available for replay, so you can refer back to it at any point. But Dr. Pittock, thank you so much for being here with us today.
Thanks very much, guys. Really enjoyed it, and hopefully see you soon.
See you soon. Bye.
Bye now.